All right, respiratory chain, also known as the ETC, electron transport chain. So, um, aerobic respiration yields a maximum of 3 ATPs per NADH and 2 ATPs per FADH. That is a test question. So during aerobic respiration, for every NADH that enters the electron transport chain, it will spit out 3 ATPs. For every FADH that enters the electron transport chain, it will spit out 2 ATPs. So anaerobic respiration yields a little bit less per each NADH and FADH. But a test question will be how many ATPs are yielded for an NADH versus an FADH. So think of NADH and FADH as the currency needed to fuel the ATP generator at the end of the ETC. We need that NADH and the FADH to enter in to wind up the ATP generator. So NADH and FADH2 are products that are made in or during glycolysis and the Krebs cycle, and they are needed to fuel the ETC. So this is the ETC, right? Electron transport chain. So it's in the cytoplasmic membrane in bacteria. So you see here are the cytochromes, and you can see you have NADH hanging out. It doesn't really show any FADH, but as each NADH comes along, it hands off the electron, and as the electron is handed off, the hydrogen passes over the membrane, and this kind of starts to wind up this possibility of, I guess it winds this system up, causing this wheel to turn, and this wheel is ATP synthase. Okay, like synthesize. ATP synthase makes ATP. That's the at the end of the electron transport chain, you're going to find ATP synthase. And if this is all wound up, and this winds everything up as we hand over the NADH and the FADH2, and the hydrogen crosses over, and the electrons help to power this. And as that happens, this gets wound up like the prop on a rubber band plane and causes the ATP synthase to start working. And as it starts working, it's basically converting ADP over to ATP, which is energy, right? And depending on if we're talking about aerobic respiration or not, aerobic respiration, as this happens, the final electron acceptor is oxygen with a byproduct of water. And if it's not aerobic respiration, then it is not oxygen. And there's some other byproducts here that I'm not too excited about. But this is the process that happens after the Krebs cycle. So glycolysis and the Krebs cycle make all these guys, right? NADH and FADH2, and along with some byproducts, some CO2, some water, some, whatever. And NADH and FADH2 make their way to the electron transport chain to do their magic, to wind it up, the ATP synthase in order to convert ADP over to ATP, which is energy. Okay, so we've made ATP in this process. So in eukaryotes, it's going to be in the mitochondria, and in bacteria, it is in the cytoplasmic membrane. All right, so let me finish this up. Hydrogen ions sequestered outside the membrane travel back into the cell using the ATP synthase complex as a channel. Oxidative phosphorylation is the coupling of ATP synthesis to electron transport. Each NADH that enters can give rise to 3 ATPs, and this is, I already mentioned this. Electrons from FADH enter the electron transport chain farther down the line. Okay, so NADH enter early, FADH enter farther down. Therefore, the FADH aren't going to give us as many ATPs. So keep that in mind. Final step in aerobic respiration. I'm not going to ask you any of this chemistry stuff, but in fact, don't even worry about this slide. Let me see what I want here. So this is important because I mentioned this before, that there are some byproducts that happen, and some cells can handle those byproducts when oxygen is used, and some can't, right? So a potential side reaction of the respiratory chain is the incomplete reduction of oxygen to superoxide ion and hydrogen peroxide. So aerobic organisms, have neutralizing enzymes that deal with those toxic oxygen products. Some do not, but aerobes do. Anaerobes do not. Aerobes do. So these are the things that they use to take care of this superoxide and hydrogen peroxide. Streptococcus lacks these enzymes, but still grows well in oxygen due to the production of peroxidase. All right. So the terminal step in anaerobic respiration utilizes non-oxygen things as the final electron acceptor. And let's see. Okay, so the only thing I really want you to focus on this one is that none of the anaerobic pathways produce as much ATP as aerobic respiration. All right, and this is an overview of aerobic respiration and what's happening in glycolysis and then the Krebs cycle and then in the ETC. So, and then the numbers are all put here so you know what is made. And then this is kind of summarized here. So I'm not going to focus too much on this, but reading through this will help, because I really try to simplify it on this chart. And then you should know how many of each are made. So use that as a reference. And this is review that during glycolysis, no oxygen is needed. Doesn't matter whether we're talking about aerobic, anaerobic, or fermentation, no oxygen is needed in this step. And remember, glycolysis happens in all three. That's probably a test question. But then once we get to the Krebs cycle, then yes, we need oxygen. We need something to fuel it, right? Even if it's just a little bit of oxygen. So in aerobic respiration, if oxygen is available, it's able to go through the process, and oxygen is the final electron acceptor. In anaerobic respiration, there may not be either not a lot of oxygen available or we'll just say not a lot. So oxygen is not the final electron acceptor. We have these other things here, and I'm not going to necessarily ask you to regurgitate them to me, but keep this handy in case you need to reference it. And then in fermentation, no oxygen is needed at all. Basically, once we get through that glycolysis, if this organism doesn't have the Krebs cycle or the ETC in its system available to it, then it uses fermentation to kind of, basically it's recycling stuff. But in the process, byproducts are lactic acid or ethanol. So, and, obviously with microorganisms, that's how they make alcohol, and they can, breads and so on, right? So, all right, so let me keep going. So this is a little bit more about fermentation. So fermentation, as I mentioned before, is the incomplete oxidation of glucose. And remember, it's because this organism uses fermentation instead of using the Krebs cycle and the ETC, because it doesn't have those available to it. It doesn't have those pathways in its cell membrane or whatever. Fermentation yields just a little bit of ATP. It's used by organisms that don't have the ETC. Other organisms can repress the production of electron transport chain proteins when oxygen is lacking in their environment and then revert to fermentation. So in this case, pyruvic acid is the electron acceptor, and it just kind of recycles that back and forth. It can continue through alcoholic fermentation or acidic fermentation, and that's up on the picture coming up. That's why it says on the left and on the right. Pyruvic acid can also be enzymatically altered and then serve as the electron acceptor. The NADs are then recycled and re-enter glycolysis, and this cycle keeps going over and over again. So you can see here, pyruvic acid, we know that from glycolysis, and then we can go this way and make an alcohol, or we can go this way and make acids. Microorganisms, the products of fermentation, I already mentioned, alcohols, acids, vitamins, antibiotics, and so on. So just a little bit here on this. I'm not going to be super excited about this, but I wanted to leave this in here. As I mentioned, if it's not a carbohydrate, if we're not talking about glucose, there's other ways to break things down. There's other pathways for lipids. There's pathways for proteins. We don't learn about those, although they are there. And so, lipids are broken down by lipases. I'm not going to go through all of this. Proteins broken down by proteases. So just kind of read through this once and just have, whoops, I didn't mean to do that, and just have it as a reference. So what's the point of all this? We break down glucose as far as it'll go until we get ATP, and then we build, or we use that energy for cell division, or we rebuild, right? We rebuild, we repair, okay? Component parts of bacteria are being synthesized on a continuous basis. Catabolism is also taking place as long as nutrients are present. And cell division takes place when anabolism produces enough macromolecules to serve two cells. So through the process of breaking down, whatever carbohydrate that it is broken down, and then the cell continues on with using that energy to build macromolecules that will hopefully, the cell will make enough to serve two cells, and that's when cell division will happen. Okay, so that's it. That is it for Chapter 7. So if you have questions, let me know, and be sure to look at all the other videos that are posted because that'll be helpful, as well as if you have questions about this, you could review like Crash Course and also Khan Academy videos on cellular respiration. So questions, let me know.